Yeah, this live session already started. Hope that you guys can hear me. Uh, let's check once. Yeah, everything is perfect. Yeah. So today uh, I will go through the cell signaling part. Hope that this will hope that hope that you guys can hear me. Yeah, exactly. I don't found. Okay, fine. Now let's turn off that. So wait. Hope that you guys can hear me. Uh, yeah, my connection is okay. Let's let's go to that screen again. Hope so. Today our target is the cell signaling. Hope hope you guys can see me. My okay, that means it's a little bit slow. Okay, fine. So this is the today I'll just explain the cell signaling part. So in that part, you can also learn about all those things, and also I'll explain the whatever existings are there. So let's start that class. I think no one in life. That's why I can wait for that. Learn about all those things, and also I explain the whatever existings are there. So let's start that class. I think no one in life. That's why I can wait for that. Learn about all those things, and also I wait. Wait. Okay, so in that signaling part, uh, what we will learn? So today we will learn about the cell communication. Let's see. Let's take a cell. We can assume this is a cell, and in that cell, uh, this is a nucleus. Let's take the another. So this is the another cell. So today we'll just explain about that in the in between this cell and this cell there is some communication there. So today we'll learn about that. So as it is the CSIR course, so mainly I'll discuss the more deeper knowledge about that and the understanding about the cell signaling. So basically we all know three type of signaling is there. One is the autocrine signaling, and whether their cell, this is a cell, and if one molecule is there and that can stimulate that this cell, the self cell. This molecule is releasing from that cell, and this so this is the molecule, and that can stimulate the self cell, that is called the autocrine. So there is the autocrine signaling, and here the target cell and the responding cell, like responding cell is the target cell and the some signaling cell, both the same, like this signaling cell and sorry, so both the cells are in same, like this is the, yeah, live, yes. So this is the signaling cell and this is the target cell, both these cells are the same kind of cell. So that is called the autocrine signaling. And then we go for the some paracrine signaling. Paracrine, whether this is a cell and we take the another cell in there in between, so in between some junctions or some channels are there, like this some channels are there in between, if that secrete some of this molecule, and this molecule is going to that region to in that cell. Let's take different color. Let's take this nucleus, this is the nucleus of that cell. So that release that kind of molecule, and that can stimulate the nearby cells. So these kind of signaling are the paracrine signaling, whether they are signaling molecule, signaling cell, and the responding cells are the differ. They are not the same like autocrine. So this is the paracrine signaling. And third one is there, that is called the, you all know about that, that is the endocrine signaling. Endocrine. Okay, let's differentiate in this. Okay, written down that. Let's differentiate. Is the channels are there, they are also part of the paracrine, or it is known as the, in more pure word, that is the juxtacrine. Juxtacrine signaling. And paracrine, there would be like nearby cells, like this is the cell, and this molecule is there, they releasing this molecule, and that molecule stimulate this nearby tissue, nearby cells. So there is no some transportation, that is the paracrine, that is the paracrine cell. Let's make it more correct. Okay, and now we are talking about the more endocrine cells. So every cell signaling is important, but endocrine is more important, whether they are, this is a cell and that can release some of this molecule, and this molecule is going through the blood, going through blood, and that stimulate the another cell. So this is the three type of signaling are there. Also you can you can say four types, but the three types are mainly autocrine, paracrine, and endocrine. In paracrine, somehow it's might be different, little bit of different of the juxtacrine, but you can make it similar kind of signaling on that. So and another two important system you have to know about that. Oh my god, why the both okay, I'll just check it. Cameo, yeah, I just removed it. Yeah, hope that in all slides, that's problematic. It should be small. Okay, let's discuss something related to the two type of molecule. one is the primary messenger system and another is the secondary messenger system. So let's write down it one by one. One is the primary messenger system. So who are the primary messenger? The like you can see they are large and hydrophilic ligands bind to the cell surface receptor. They are integral membrane proteins that are embedded in the plasma membranes, and while they are hydrophobic molecules like steroid, retinoid, or thyroxine can easily cross the plasma membrane and bind to the intercellular receptor. That's the ligand is called the primary messenger. Like large and hydrophilic ligands bind to the cell surface receptor. You can understand there's a cell. I cannot go through the to more details because it's very easy thing. Like this is a molecule and this is a cell. Hope you guys can see me see that screen. So this is a large molecule or hydrophilic ligand bind to the cell surface receptor. Let's say this is a receptor that bind to that the cell, and these receptor present in the plasma membrane, and they are hydrophilic molecules like some of the steroid, retinoid, thyroxine easily cross and it's enter to the cell and bind on their specific receptor. That is called the primary receptor, or sorry, primary messenger. Let's talk about the secondary messenger. Secondary messenger, it means secondary messenger. So in the secondary messenger system, how it is there, like within the cell, one location to the another location, and result there the certain changes happen in the gene responding cell. That type of cell, like kind of the binding of ligand to its receptor leads to generation their secondary messenger system. Like you can see this is a one molecule stimulated and that molecule influence the other molecule, and this molecule going to the nucleus and changes the gene expression. That kind of molecule is a kind of secondary messenger. So hope you guys can understand that the primary messenger system, secondary messenger, and the autocrine, paracrine or juxtacrine, along with that the endocrine system. So these are the basic, all those basic things. Now we will talk about the ligand receptor binding, like how it is possible to the one ligand to the another binding to that receptor. Let's say you take one example, you can take the growth factor and other things. I hope that in few class back I have just discussed about that. These are some of these molecule, endocrine molecule that can bind with their specific receptor, like growth hormones, and I think particularly as I can remember that this I think in insulin class I have just discussed it. So now our like ligand receptor complex or ligand receptor binding, and then the receptor affinity, like receptor are said to be the high affinity, like they can bind with their specific uh, zone or specific pocket of that particular protein. So they are highly specific and they are occupied in low concentration of this receptor, and similarly the high concentration of ligand is required to its receptor become occupied. So whether there are ranges of some of these important constant value like called the dissociation constant or dissociation, or this known as the kd value, the receptor affinity. Receptor affinity denoted by the kd value. There is a kd value like that is called the dissociation constant, that is known as dissociation constant. So this is a dissociation constant, that is the known as the Kd. There is the Kd, and that denote the receptor affinity of that. So what is the value of ranges in between them? So you can assume about the 10 to the power minus 7 to the 10 to the power minus 10 molar. So that kind of value, the receptor affinity is such ligand has a very veil of this Kd value. Likewise, their low affinity receptor possesses the high value of the Kd. That means they are, you can say, the low affinity. I cannot get the good color. Low affinity. Yeah, that is the low affinity. Low affinity that possesses the high value. High value of Kd, dissociation constant. So basically in some questions they are kind of like comparing this value. So comparing this kind of value, and that will be like in that value it is like uh, so in some of these mathematics, like they are low affinity, it means their binding is very much minute, and that time the dissociation constant will be more. That's a basic thing, but in some questions it will be like they give some value and ask about that, the which molecule it can they are in dissociation part, which will be like more stable or less stable. That kind of questions are coming, and only you have to know about that the affinity and relations with the value. So they are kind of inversely proportional. Their affinity is low, that means high value of the Kd. So you have just like L uh, proportional 1 by Kd. Yeah, you can think about that easily. Now now there another important key factor is the receptor down regulation. Like, what is the receptor down regulation? The cell uh, possesses the ability to sense the changes of its ligand concentration, and whether receptors are occupied by ligand or prolonged period of the time. Like there is a cell surface, let's say these are the receptors, this is a cell surface, the receptors are there. So like now I go through these are called the ligand. So these ligands are there. So if their concentration get decreased or diminished, their cell can sense that kind of changes of the ligands concentration, and if their receptor is kind of occupied, if a prolonged period of the time, the cell adapts itself. So adaptation happens, and for that the no longer responds to that ligand. So to stimulate the cell further, the concentration of the ligand must be increased. So let's say it's diminished, but and these are completely occupied. But if you want to more want to just restart of that system, so that for that you need to changes the concentration of that specific ligand. So that's why it's bind to that specific ligand, and then their receptor down regulations will come. So that's why it's called the receptor down regulation, and there are such changes, it's known as that regulation. And another important factor, you just know about the receptor mediated endocytosis. Their cells can change the number of the receptors. Like, let's say it's a cell, it's a cell membrane, and receptors are there in that specific position, and whether they are like cell, this kind of receptor, it can be like endocytosis happen, then the receptor are going to inside. That means the surface receptor will decrease. Like Okay. Okay, so that's kind of the like surface receptor get decrease, and once a time it will fill. So for that, it's kind of their removal of the receptors from the cell. So that is the known as the receptor mediated endocytosis. So these are the receptor mediated endocytosis, and this previous one, and that is the called the receptor affinity. That is the receptor affinity. So in that class, in that live session, I'll discuss about the cell signaling part. Already we have discussed the uh, autocrine, paracrine, and the juxtacrine, and along with the endocrine signal. And it's a very little bit. And then we have discussed some of the primary molecule and secondary molecule. Now we are discussing about the ligand receptor binding and their affinity, and then we go for their receptor affinity like dissociation constant like Kd, and like their value will 10 to the power minus 7 to 10 to the minus 10 molar, and these has some relations with the dissociation constant. If in the high value of the dissociation constant, that will give you the low affinity. And then we are discuss about the two thing, one is the receptor affinity, another thing is the receptor mediated endocytosis. If the receptor affinity like their concentration of the ligands that can bind with the specific receptor, if they are all are like ligands are bind with the specific receptor, that time it might happen there some signals are not coming to later or downstream. That's in that time their concentration of ligands should be increased. At that point their system getting recovered. So and then like another kind of mechanism are there. Let's go for that. So another kinds of mechanism are there, like that is called the desensitization. So one mechanism we have already, the receptor down regulation. Already we have discussed about the down regulation, and another is the desensitization, and which includes the changes in the receptor, and that decrease the affinity. So changes in the receptor affinity will decrease for this ligand and render unable to initiate the changes the cellular function. That is called the desensitization. So you can just remind it, desensitization. So that is a desensitization in that mechanism. So like decrease the receptor that decreases its affinity to the ligand, and that's why the cellular changes has not happened. So that is the desensitization mechanism. So like you can briefly write down, and that cannot be notes. Okay, fine. Changes in the receptor, you can. This is the changes of the receptor, and that decrease the affinity, decrease the affinity to the ligand, and then they are rendered, they are unable to initiate the changes in the cell function. So that is called the desensitization. And another two important key things like agonist and the antagonist, like some of the synthetic molecules are made and that can bind specifically to the particular receptor, and these are used as a drug and the treatment of many medical problems. And if they activate the receptor, what happened to this camera? So if activate the receptor, these are called the agonist, and if they are it inhibits the receptor, that will be the antagonist. So you just write down some points like these are kind of synthetic ligands that can bind specific pocket or specific protein of this particular receptor and stimulate them to downstream process, then it will be like agonist. And if they are inhibits their activity of this specific receptor, that is called the antagonist. So agonist and the antagonist. So both the term, you hope that you understand this. Now the signal integration part. So what is the integration of that signal? So different signaling pathways that can operate at a given moment of within a cell. So the component of this pathway interact with each other and also affect each other in the transmitting cellular responses. Sometimes one receptor can activate the multiple pathways upon the ligand binding. So that generally seen that the multiple pathways are activated in the similar time or simultaneous time. So, in some cases, the different ligand bind to the different receptor present in the cell surface of same cell and activate the specific signaling pathways within the cell. So, these are called the like cell integrate all of these signal to generate their coordinated responses. Like in a school, like the different departments are there, but their operation in the same time that can operate like a single molecule that can stimulate the different kinds of pathways in a single time. That might be, you can think, the lot of functions controlled by a single function, like they are single function controlled, they are different kind of function in simultaneous way. So that's why this is a kind of mathematical problem is there. You can identify this problem and you can specify these functions and which signaling molecules or signaling pathways that can show their more prominent way or of the downstream effect. So that's why it's kind of signal integrations, like one single pathway or that can control or activate their multiple pathways, activate the multiple pathways. So now we are already discussed about the signal integration. Okay, so now we are discuss about the signal amplification. So what is the signal amplification? Like activation of the single cell surface receptor molecule by ligand can result the stimulation of the large number of secondary messengers. So each of these in turn activating its target protein affects the activity of multiple downstream pathways, and many signal transduction pathway amplification is much more necessary of the density of the receptor. So you can think uh, like signal is amplified by different kind of pathways and they sustained with it and then affect the downstream pathways. So that is the signal amplification. So hope you guys can understand about that, the like amplification of that signal. You can also say that the also in previous you can see the integration part. Now let's talk about some of these understanding, like okay, so like one molecule we is called the epinephrine. epinephrine. So this is the molecule, and its binding is shown the 10 to the power minus 10 molar. So already we know about the Kd value. So like m 10 to the power minus 10 m is their epinephrine. So in that epinephrine, okay, spelling is right. Okay, now it stimulates the one of the important molecule that is called the adenine cyclase AC. So adenyl cyclase is activated, and adenyl cyclase that can stimulate the CAMP, cyclic AMP, and this cyclic AMP is the 10 to the power 6 m. Now this CMP stimulate the another important protein that is called the protein kinase. Now you can see their epinephrine, one epinephrine that made the kind of adenine cyclase, and this adenine cyclase stimulate the CMP very much, and then one individual CMP that can activate the some protein kinase, and this protein kinase the kind of single molecule, and then that activated the enzyme, and this enzyme to the product. So that's why its kind of signal is getting amplified in broader nature from a single molecule to like tree like branches, and it gets a more amplification. That's why it's called the amplification of the signal. Hope you guys can understand with my live session, and I'll go through some another 10-15 minutes, 10 minutes you can say. Okay, let's delete that, this slide. Okay, let's delete that. Yeah, now I will go through the another important key aspect of the G protein link type of receptor, and some of the activation of that specific ligand and their structure of the specific protein. I'll not go through too much details, but still I want to cover with that with very minute details like G protein. Let's draw it. No, this is another class is coming now. ah G protein linked receptor, kind of G protein linked receptor. So in that G protein, are because of their ligand binding, they undergo their G protein is guanine nucleotide binding protein. So you can write down here guanine guanine nucleotide binding protein. So in that protein, some of the example like G protein linked receptor are like olfactory receptor, some of the norepinephrinic receptors, or some of the hormone receptors that might be like narcotic drug like morphogen binding opioid receptors. So which are the G linked protein receptor? So G linked protein receptor, so like their G-link protein receptor have the 7 transmembrane domains are there, like 7 alpha helices are there. Like you assume this is a cell membrane. So these are the, you can say, transmembrane. So let's write down the 7, 3, 4, 5, 6, 7. So these are the 7 transmembrane protein. It will be like connected with each other, like connected with each other, right? And then it will be connected in here, and then it will connected there, and in that way it will be like that. Let's say that is a C double O minus, and their top part it will be like NH3 plus. So this is a 7 transmembrane protein. So like G protein coupled receptor, their N terminus will be in the cytosol. Sorry, it will be in their extracellular fluid EC F region. Let's write down clearly. That is the extracellular fluid. That is the extracellular fluid, and this is the part of the cytosol. Let's making it more relatable. So this is the cytosolic part. Yeah, this is the cytosolic part of that, and this top part is that. Okay, now let's, this is the cell membrane part, and make it more that. Yeah, so this part is the outside of that cell. So in that transmembrane protein, how that you get that structure easily, there that will 7 transmembrane protein. These are the alpha helix are there in the transmembrane protein. So transmembrane protein, okay, so hope that you guys can understand. There are 3 loops, or like this loop and these 3 loop, these are inside of the cell, and these 1, 2 and 3 loop in the outside of the cell. These are NH3 domain, and these are C double minus domain. Now this receptor, like the G protein receptor, are can be regulated most of the common mechanisms, and regulation by the phosphorylations of the specific amino acid residues located in the cytosolic domain. And if they are like kind of phosphorylated, the phosphate group is bind with that, and then like this protein receptor like GRKs are getting activated with the G protein coupled receptor, and another type of kinase or like protein kinase A, which itself activate the G protein coupled receptors for their signaling. And sometimes their regulations, kind of negative regulations are also happen. Hope that you guys can understand my point of view. Like in the cell signaling class, for still now what I have discussed about the autocrine, paracrine and endocrine signaling, and then we have discussed of the some of the receptor properties, and in also the affinity, receptor affinity or dissociation constant, that is a KD, and their normal range of the value, and their relationship of that, like if their KD value is more, and that they are inversely proportional to that their like affinity of this receptor, and that and also the down regulation, agonist of this receptor, antagonist of this receptor, and signal integration, and with the example with of epinephrine to the signal amplification. And then we just going to the G protein linked receptor or coupled receptor. So in that receptor already we have known about the 7 transmembrane proteins are there, and these are the alpha helix, and that bind with that. Hope that in the screen you can understand, you can see. And this cytosolic part, in cytosolic part COO minus group will be there, and NH3 group will be in that cytosolic part. Now the structure of the G proteins. Oh, sorry. Let's take a new slide. Insert cameo slide. Let's delete that and making it small. Hope that you guys can understand. So now we'll go through the G protein and their structures. Like G protein, okay, let's make that slide. Let's one time. Yeah, Control D. Yeah, hope it can help us. So let's see. Yeah, now it's fine. So draw. let's say let's say this is the switch on. G protein is like kind of the molecular switch, on and off. So its states depend on the GTP. The G protein is bound to the GTP or the GDP. So two molecule, you have always remember GDP, and another molecule is the GTP, guanosine triphosphate and diphosphate. So these two type of G proteins are in the large heterotrimeric G proteins of small monomeric G proteins. So their subunits are like, acha, on and off. I'll just complete it, that if that is a good thing. GDP and GTP. So these are two molecule that controls G protein couple receptor or ligand receptor. G protein couple receptor. So like off or on, that depends on that. So it has three subunit. One is called the G alpha subunit, another is called their G beta subunit, and the another subunit called the G gamma subunit. And all the heteromeric G proteins have the same basics of structure. It might be some of these difference are there, like this basic structure are kind of same. So in that structure, like these are the G alpha. This is the more largest. This is the largest among all these subunit, and it can bind GDP and GTP. So it can bind both. But when G alpha binds with GTP, its affinity for the G beta gamma is decreases, and therefore separate the G beta gamma complex. So if like G alpha bind with ATP, then beta gamma complex separates, and like their affinity decrease from the G alpha complex. But G beta and gamma subunits don't have their guanine binding nucleotide binding site, and they remain permanently bound to each other. Like beta, that's why I'm writing down that the beta gamma complex. So they don't have the guanosine binding complex. Only binding complex is there in the G alpha domain. Now an active on form the bound GTP, and inactive form of that will be the off state. Like you can write down on, this is off. Clear? So now we will go through the another important factor, that is known as the GEF, guanine nucleotide exchange factor, which cause the release of the GDP from the G protein, because it's in inactive state. If the GTP bind with the G alpha, then it's inactive state, but it's need to be released. So for that releasing purpose, this is necessary, that is a GEF, guanine nucleotide exchange factor, GEF. At that time it will be active. So another 2-3 important factors are necessary Like one is the GTPH activating protein, that is known as the gap protein, and regulators of G protein signaling, that is the RGS signal, to accelerate the GDP hydrolysis. So let's write down some of these mechanism of that. Let's erase something, or it might be smaller. Let's try. Okay, now making it small. Yeah, now we have a space. So let's write down of it. So like G alpha subunit, G alpha subunit with the, let's say it's a binding pocket. So in that binding pocket, you say the GTP is bind there. So GTP already we know that the it's a on green. It's on. Now you write down the G alpha protein and you bind the GDP. If the GDP is there, that means it's in off. Now how it will be the exchangeable from the GDP to GTP. So how it will be possible? So some of the activator proteins are there. So you said that some of the activator protein. So if it is the GEF activator protein, guanine nucleotide exchange factor, if it is there, and what happened? GDP is released and GTP it's bind to that region. Hope you can understand. So this is the activator, GEF, guanine nucleotide exchange factor. And if the activation is there done, if the gap is there, gap, GTPH accelerating protein. So this is an inactivator protein and that release the 1-phosphate, and if release of 1-phosphate, that will bind the try to diphosphate. Hope that you guys can understand about the activation and inactivation of the G protein. So this is the activation and deactivation. Okay, for some important call, I just leave that streaming and hope that after that I'll continue.